So the caption is, true, my last several experiments fizzled and a year's work has gone down the drain. But I'm not discouraged. My video game, Antigens and Antibodies, is selling like hotcakes. I'm not old enough to remember hotcakes as a thing. It's an expression. I'm not entirely sure what a hotcake is. Is it a type of pancake? Is it heated up dessert? I couldn't tell you. And I haven't bothered to look. You could probably look it up though. As we often do, we'll add some stuff from the last video. This is just a very abstract representation of the molecules, where you've got the neurotransmitters that attach to the receptors. This impulse comes down here, causes the release of neurotransmitters. If enough receptors pick them up on this end, the impulse continues, as it activates the depolarization of the membrane on the other side. All proteins. This shows the quaternary structure of an antibody. This light pink stuff and the light green stuff are stable from one antibody to the next. These darker areas are variable because it needs to be able to fit. This is the antigen binding site. So it needs to be stereo-specific to fit whatever it is it's made to go against. Now, from what I've been reading lately, we have these pre-built for an amazing array of potential antigens already embedded in certain, what they call antigen-presenting cells. So I'm got to do some reading. Immunology has gone way past my immunology class that I took many, many years ago. So I'm myself starting to learn new stuff about this. Is that reading about COVID has brought me up against a lot of newer concepts in immunology. This is supposed to be a representation of the inside of a bacterial cell, showing the molecules. I think it would be both sparser and more crowded than this. There'd be more molecules, and also there'd be a little bit more space amongst them, because even in a bacterial cell, you can't see the molecules particularly floating around in there. But this ropey stuff down here is supposed to be DNA coiled around histones, which is a type of protein. Remember I said the protein list was longer than we talked about? This is the membrane with, this is the part of the cell wall up here on the outside of the membrane. This is a bacterial flagellum, which interestingly enough is one of the few examples in nature of a true wheel, is that this thing spins the flagellum with the same way that we have a wheel, where we have a column and then interactions are in a disc around it makes the thing spin. Our flagella don't do anything like that, but bacterial flagella do, work in a very different way, not homologous. And it's just supposed to show you a lot of stuff. A lot of stuff. So now we're going to the last group, the nucleic acids, which you've all heard of even if you don't realize it, but you'll realize it when I tell you the initials work, Na, D, Na, deoxyribonucleic acid. The two vaccines that are out, messenger RNA, ribonucleic acid. So these are things that we kind of sort of know without necessarily having it in our head what the initials stand for. So these are also polymers. So we've got three major polymers we've talked about. We've got starches, which are polymers of sugars. We've got proteins, which are polymers of amino acids, and we have nucleic acids. The bits can be called nucleotides, they can be called nucleic acid bases, they can be called nitrogenous bases. I do find that it can be a little confusing when people are first getting into this, where you talk about nucleic acid bases. But what it means is the whole molecule behaves differently than the bits and pieces do if you break the bits and pieces off. Break the bits and pieces off, they act like bases. In a string, they act like an acid, right? Because it all has to do with what you do to your surroundings. If you're raising the hydrogen ion concentration, then you're behaving acidically. If you're lowering the hydrogen acid concentration, you're behaving basically. This is a nucleotide. It has a sugar component, in this case deoxyribose, that's where the D name comes from. It's got a nitrogenous base, very much related to an amino acid, but it's not an amino acid. And it's got a phosphate group. And there's a limited number of these things. Well, again, it's not a limited number so much as in living things there's a limited number. I'm not going to expect you to remember the purines and the pyrimidines, but that's what your two groups are. You've got A and G. They are short. And you have, no you don't, sorry. Those are the long ones. The pyrimidines are the short ones. And they cross-connect to the other side. So this long one connects to a short one over here. This short one connects to a long one over here. Adenine connects to thymine. In some circumstances, connects to uracil. Guanine connects to cytosine. So I'll write that all down in a second. Later on, when we talk about replicating DNA, we talk about building, we take and pull the two strands apart. This is one side of a DNA molecule. Pull the two strands apart, and what's on this side, when you're building a new one, determines what's on that side, because there's only one match on the other side. But mistakes get made, and if you put a short one and another short one over here, then the two sides collapse in a bit. If you put a long one and another long one, the sides go back out. And one of the simplest proofreading molecules tracks along the newly built DNA, and if it hits a dent, it grabs the enzyme that makes it, pulls it back, goes, okay, try again. If it hits a bump, it does the same thing. So adenine pairs with thymine. You've got all these molecules laid out. There's links in the book to show you what they look like. And I'll put something up here. Uracil is, RNA doesn't typically have thymine. It's not thiamine, which is a vitamin. That's THIA-min. RNA has uracil, and it looks very similar to thymine. And cytosine pairs with guanine. So if there's guanine over here, there's cytosine over here. If there's cytosine over here, there's guanine over here. In DNA, if there's an A over here, there's a T. If there's a T, there's an A. And the way I learned it, just very, very quickly, is to put them in alphabetical order. And remember that the inside ones pair together and the outside ones pair together. So C goes with G and A goes with T, or U, right? Still works. And DNA produces what are called complementary strands. Now, if that was complementary with an I after the L, that would mean they were, giving each other compliments, they were buttering each other up. Complementary strands means that if you know what's on one side, you know what's on the other side. They don't match, they're not matching strands, but they're complementary strands. And that makes it easier to pull strands apart and build in what used to be there pretty reliably. It is possible to build two-stranded RNA. I think that's what they're doing with the PCR tests for the COVID virus, but I'm not 100% sure of that. But RNA is typically, in most natural systems, is a single strand. Now we're going to follow up on a lot of this later. And you've covered some of this already. A gene is a code, not for a trait, not for passing on to offspring. A gene is a code for one particular type of protein. So there's a gene for insulin, there's a gene for glucagon. There's a gene closet for antibodies, because you can make a whole bunch of them using different bits from the gene closet, right? That's what happens when you have quaternary structure. You can actually build different things with different bits. But every protein that we make has a gene for it. This is way more complicated than that. It's still introductory here. There's going to be some things you're going to find out later are only sort of true. If you have a gene for a particular protein, it's a long sequence of nucleotides. If you find a version of that gene and the sequence is different, that's an allele, or an allele. I've heard it pronounced both ways. Is that in a lot of cases, a different allele doesn't really change the protein product at all. Is that the translation system is what they call very redundant. Is you can mess with it in little ways and get the same product you were going to get for a number of different reasons, which we'll go into when we get into the nuts and bolts of the system. But in some cases, a different allele will produce a different protein function. Most of the time, if you change the sequence enough that it changes the function of the protein, the likeliest outcome is the protein isn't going to work, or it's not going to work as well as it should. But sometimes you get it and it does something totally different than it was supposed to. Kind of cool. A codon, we talked about this before. Every amino acid in the protein sequence is coded for by 3 nucleotides along the DNA sequence. Those 3 nucleotides are the codons. Technically, the codons are in the translation molecule in RNA, but it's still a 3 nucleotide to 1 amino acid ratio. Again, something we'll cover later. We've got a lot of DNA. I mean, literally the DNA in each one of your nuclei, if you stretched it out, would be this long. So it's shoved into a very tiny room. You need to be able to package it really well. And so you combine the DNA with those spooling proteins that they showed very simply in the overhead that I had up a few minutes ago. And they spool and they pile and they clump. And that's a complex. It's a DNA protein complex, and that's what a chromosome is. And we don't keep all of our genes as separate pieces of DNA. It would be impossible to copy and distribute those when you made new cells. You'd lose track of them because there'd be thousands and thousands of them. So to reduce the number of movable DNA pieces to something that you're not going to screw up, we put a bunch of genes on one long connected piece of DNA. that's a chromosome. And again, all stuff that we'll talk about later. But the basic molecules, the basic organic materials, that covers that for this chapter.